Good morning everyone. My name is Yasmin Villa and you are welcome to Plutus IAS. Here we have started a series to discuss previous year questions with you. Today I am here to discuss science and technology questions with you. Without wasting time, let us start with our first question. And the first question says, consider the following activities: spraying pesticides on a crop field, inspecting the craters of active volcanoes, collecting breath samples from a spouting whales for DNA analysis, and at the end of these statements we have a command: at the present level technology, which of the above activities can be successfully carried out by using drones? Here first we should know about the drones, the basic part. The static part. Drones means unmanned aerial vehicles. Clear? We are having number of
जी मैम वो दिक्कत आ रही है इस बार वो कैमरे में तो वो इधर उधर हुआ तो वो ब्लैंक आ रहा है कैमरा। मैं चेक कर रहा हूँ। मैम एक मिनट चेक करता हूँ। मैम चेक कर रहा हूँ अभी। एक मिनट मैम चेक करता हूँ। मैम कैमरा चेंज किया हूँ अभी। मैम एक बार डेमो लिया हूँ वो हुआ नहीं।
hello. Because of some technical problem, we are running slightly late. So let us focus on the given question. Here the question is about the drones. Drones are unmanned aerial vehicles and DRDO manufactures some of the drones named Lakshya Nishant. Drones are used for spraying pesticides in the agricultural fields. This is a right statement. But from the year 2020, our government has declared this illegal to spray pesticides using unmanned aerial vehicles. There is one legislation, the Insecticides Act 1968. This Insecticide Act 1968 has a provision that you cannot use those methods which are harmful for the large number of people. So while you use drones, you are spraying pesticides in the air, and it can migrate to far away distances. Because of this reason, the government prohibited the spray of pesticides using unmanned aerial vehicles, but we are able to do that. It is asking you can be. Can be means the probability, so this statement is right. Then the second statement, craters of active volcanoes, without any doubt, this we are using. In the year 2015, some scientists in the Hawaii island, they used unmanned aerial vehicle to take samples from an active volcano. So this statement is also right. For this question, for every question in the science and technology, you require a mix of both static knowledge as well as the dynamic knowledge. And here, along with the static knowledge, what are drones, you require some additional part which is on the basis of current affairs knowledge. This is also right. Then comes the third statement, you can collect breath samples of the whale. This is also right. Reason being, in the year 2015, we again had one more research paper in which some scientists in the South Africa used drones to collect and to monitor whales. So this statement will also be right. Now, after knowing about all the three statements, we can easily come on the answer, and the right answer for this question will be D, 1, 2 and 3. Let us move on the next question. The experiment will employ a trio of spacecraft flying. Trio means three. Trio of spacecraft, it means three spacecrafts flying in formation in the shape of an equilateral triangle. It is saying three spacecrafts moving or making a triangle in the space that has sides 1 million kilometer long, and one side of this triangle will be 1 million kilometer long with lasers shining between the craft. It means they are making a triangle, equilateral triangle. These spacecrafts are making an equilateral triangle in the space, and length of one side will be equilateral, means all the three sides equal. Length of that equilateral triangle will be 1 million kilometers in the space. This experiment refers to, before this we must know one more experiment, LIJA Pathfinder. What is the full form of LIJA? This is Laser Interferometer Space Antenna. What is this? Laser interferometer. Space antenna. Basically, this is an experiment to study gravitational waves. So, this earlier this was named LIJA Pathfinder, but this sky version of this LIJA, Laser Interferometer Space Antenna, is the evolved LIJA. This is one experiment to study the gravitational waves. How we define gravitational waves? Gravitational waves are the disturbances in space-time fabric. This is how we define gravitational wave. To study this disturbance in the space-time fabric, one experiment was designed, and according to this experiment, laser interferometer space antenna, now we made a triangle in the space to study any change in this fabric, in this space-time fabric. Why there will be any change if there is any event in the space, for example collision of two massive bodies like two black holes or massive explosions, during those processes there is a release of high amount of energy as well as the gravitational waves. To study those gravitational waves, this experiment of making a triangle in the sky is known as evolved LIJA. Clear? Now, why we did this will study the gravitational waves. Though this was on paper only, still it is not launched in reality. It will be launched in the year 2034. It will be launched in the year 2034. So you must know about this. Now the next question. Next question is, in India the term public key infrastructure is used in the context of, please remember, first you need to read the question very carefully. Reason being, a question can have some hint for solving it. Clear? Public key infrastructure, public key will be like a password. This is in context of the digital signature. What are digital signatures? Digital signatures are the equivalent versions of the manual or physical signatures. This is recognized under the IT Act 2000. Under the IT Act, there are certifying agencies which provide digital signatures to individuals. Those digital signatures works through public key and the private key. What is the public key and the private key? Public key means this is the password or the key which is published by the certifying agency in the public, for the public. For example, if I am a person who is certifying or who is using his own digital signatures, then I will be encrypting my content into the digital form by using a private key. That private key I will be getting from the certifying agency, and this key is nothing but a password. This key is known to me only, but when I send my email or when I send my document to another person, that person will come to know that this is coming from me. How he can authenticate about this document by my digital signature? How he will be using it? He can go over the internet, and on the internet he can check the public key about me. If he will get all the information that public key is issued to USB Rohila by this company on that date, and it will be valid for that day, then he can take that public key. He can use that public key to decrypt my sent message. Clear? So public key infrastructure, this whole setup is known as the public key infrastructure, and this is a term used in the digital security infrastructure. Next question. This is our next question. It states, in the context of recent advances of human reproductive technology, pro-nuclear transfer is used for. This is a straightforward question. If you know pro-nuclear transfer, you can easily find out the right answer. Pro-nuclear transfer means, what is a, first you must know the pro-nuclear, then it will be very clear to you where it is used. Pro-nuclear means during the fertilization, like the formation of the zygote and the embryo, we are talking about the reproduction. In the reproduction, one male gamete fuses with one female gamete, and both results in the formation of a zygote. Now this zygote will be transplanted, and this zygote converts itself into embryo, then into the offspring. This is the complete process. But what happens in some pregnancies or in some offspring, there is defect in the mitochondria, and we know during the fusion between two gametes, the egg from the female fuses with the sperm of the male. So ultimately egg is having the complete setup. We know the structure of a cell. Cell is having a nucleus and different organelles, cell organelles present inside the plasma membrane of the cell. This is the structure of the egg, and in this cell the sperm which will penetrate inside it and fuses with the nucleus. Now ultimately this cell which is having egg inside it, so this cell will transform itself into the embryo and the offspring. So in every child the mitochondria is coming from the mother, but in some female this mitochondria is defective. So what doctors do, they go for mitochondrial gene therapy. What is this? In the mitochondrial gene therapy, they take out one egg from a third female. In that egg, they will remove the nucleus of this egg. What they will do? They will fertilize sperm with the egg of original mother, and in the laboratory, after doing this, they will be taking out this fused nucleus. In this nucleus now sperm has penetrated and sperm has fused with the nucleus of this egg cell. After this fused egg cell, we are taking out this fused nucleus of the egg cell. This fused nucleus is known as the pronucleus. Now we are removing this pronucleus from the egg cell, and we are inserting this nucleus into a third lady, like the third lady from where we have taken out the egg cell. We are inserting this pronucleus in the egg of the third lady, in which we are having healthy mitochondria. Clear? Because there was a defect in the mitochondria of the original mother, we have taken egg from a third mother and we have inserted this pronucleus inside it. Now this zygote will not be having defective mitochondria which was present in the original mother. So now this baby will be healthier. So this is a mitochondrial gene therapy. In the mitochondrial gene therapy, we use pronuclear transfer. This is a way we make healthy babies. Now this is also one of the example of the three parent baby. Please remember this term. So this is a term used in the context of prevention of mitochondrial disease in the offspring. Here the statement D will be the right answer for this question. Let us move on the next question. This question says, with the present state of development, artificial intelligence can effectively do which of the following? Question is about artificial intelligence. So first we must know about the artificial intelligence. It is the intelligence shown by the machines. Now let us look on the options. First option says, bring down electricity consumption in industrial units. Is this possible or not? Yes, it will be possible, because we are using AI for planning, for operational things. If we can plan in a better way, it means we can decrease the electricity consumption, so it will be fine. First statement is right. Second, create meaningful short stories and the songs. We are using Google's for it. Google has different AI product like Google Art. In that you can do all this, creating short stories and the songs. Disease diagnosis, yes, you can do disease diagnosis else also. This was also demonstrated by the Google. Google made one neural network, artificial neural network. By using that artificial neural network, they tried to find out the cases of lung cancer, and it was observed that this was much more efficient than any doctor. So you can do disease diagnosis. This is also right. Next, text to speech conversion. We are using it. Number of chatbots are there. We are using on the daily basis. So it will be right. Next question. Next option: wireless transmission of electrical energy. This is also a right statement. Reason being, we are able to transmit electrical energy by using artificial intelligence. We have seen examples like phone charging, laptop charging through the airless transfer of electrical technology. That is possible only with the help of AI, artificial intelligence. So all the 5 statements are right in this question. So the right answer will be 1, 2, 3, 4 and 5. Let us move on the next question. With reference to VLC, VLC means visible light communication. Which of the given statements are correct? Let us first discuss about the VLC, visible light communication. This is a wireless data transfer technology. In this wireless technology, we are using visible light. We know the visible light is having a wavelength of 375 nanometer to 780 nanometer. So we are using this wavelength of light to transfer the data. Yes, we can use it. In 2011, one scientist named Horace Hall, he discovered that we can transfer data by using light, and this technology was given a name light fidelity, Li-Fi, L-I-F-I, Li-Fi. Li-Fi is nothing but light fidelity. Harald Haas was the main pioneer of this technology. What he did, he took one LED bulb, and below the LED bulb he put one laptop. What he did, in the LED he implanted one data processing chipset, and he tried to transfer data from this LED bulb light to the nearby laptop. He found that he was able to transfer the data between the LED bulb and the computer system he was using. He later tried to block the light. He found that the signals blocked when he tried to block the light. So he concluded he can transfer data with the light. So by this way, this is also true that VLC can be a technology for data transfer. What he found, the speed of data transfer is very high when you are using visible light. It can be into like around 224 Gbps. 224 Gbps is much more than the available wireless technologies like the Wi-Fi direct, Bluetooth, clear. So you can use it, but there is a problem that this is only possible to transfer data only up to smaller distances, not far away distances, because visible light cannot travel through the physical objects or the concrete structures. Because of this quality, the data communication can take place only up to a smaller distances. Clear? Now let us look on the various statements. VLC uses electromagnetic spectrum wavelength 375 to 780 nanometer. This is a right statement. I already told you. VLC is known as long range optical wireless communication. Long range cannot be there. I told you, if you obstruct the flow of light, it will also obstruct the flow of the data. It means it cannot be for the long range. Second statement is wrong. See, now if you want to solve this question and you are attentive to the given options, you can easily find out that only one option among the 4 options is not having the second statement. This will be your answer. If you are attentive while solving the questions, sometimes you can save the time, you can save your efforts. I am very sure that second statement is wrong. Now, as per this information, only C option is there which is not having the second statement. All the other 3 options are having the second statement. So my right answer will be C only. But let us look on the other given statements. VLC can transmit large amounts of data faster than Bluetooth. Yes, I told you this part. VLC has no electromagnetic interference. This is also true. Why? Reason being, this is for a short distance, and there are very few frequencies which can intermix it. So in practicality, there is no electromagnetic interference. Interference means mixing with other signals, mixing with unwanted signals. That does not happen in the Li-Fi or the visible light communication. So this is also a right statement. So the right answer will be 1, 3 and 4. With reference to the carbon nanotubes, consider the following statements. Before going forward, we should know basics about the carbon nanotubes. Carbon nanotubes are the cylindrical structures of the carbon, and carbon nanotube is an allotrope of carbon. Chemically, it is an allotrope of carbon. Allotrope means the substance which can have more than two or more than two physical forms. Clear? So this is an allotrope of carbon, and this is in the cylindrical shape. The main peculiarity about carbon nanotubes is they are modified form of, you can imagine them as the modified form of graphene. Graphene is a two-dimensional allotrope of carbon. If you turn this into the form of a cylinder, you will get a carbon nanotube, and this carbon nanotube can be connected with number of other things. Clear? When you connect it with number of other things, you can use it for different applications. For example, I can conjugate means connect it with the drugs. Clear? Drugs are the molecules only, we can connect both. So you can use carbon nanotubes for targeted drug delivery system. Second, you can also use it for certain diagnosis. You can use it as a sensor. How sensor? You can attach different chemicals like different amino acids, proteins with the carbon nanotubes. They are of very small structure. The size of the carbon nanotubes is only 1 atom thick cylindrical structure. So you can imagine they can travel anywhere inside your body. So this is the added benefit. Because of this, you are having number of medical applications. One most important thing about carbon nanotubes, if a question comes, it is a one-dimensional, two-dimensional, three-dimensional. Right answer will be one-dimensional structure. How? Carbon nanotubes diameter is 0.001 nanometer. I am talking about the diameter. And if you look at the length versus diameter ratio, it will be like 13 crores 20 lakhs. 13 crores 20 lakhs is to 1. If it is having 1 nanometer of diameter, then length will be 13 crore 20 lakhs nanometer. Are you understanding? Like very long in size but very small in diameter. When you look it at from a reasonable distance, you cannot look over this with the naked eye. We are doing all this in the electron microscope. So whenever we are doing this in the electron microscope, you can imagine it. Now it will be looking in a single dimension. So whenever a question comes, it is a single dimensional structure. Please remember, yes, this is a right statement. Though this is in a cylindrical form, but this will be a single dimensional structure. Let us look on the statements. They can be used as carriers of drugs and antigens in the human body. Obviously, I told you how we can use them. They can be made into artificial blood capillaries for an injured part of the human body. Yes, this is also possible. Reason being, these are cylindrical structures. In the cylindrical structure, inside it we are having the hollow space. This hollow space will be can act like a capillary. So we can use it like a capillary in the body. Next, they can be used in biochemical sensors. Yes, we can convert them into the biochemical sensors by conjugating it with certain other chemicals or certain other substances. That is also feasible. Carbon nanotubes are biodegradable. Yes, obviously we are studying about the biodegradable substances. All the carbon compounds, organic compounds, they are biodegradable. What are organic compounds? Those substances which are made up of carbon. So carbon itself is a biodegradable substance. This will be right. So all the given statements are right here, and the right answer will be D. this is our next question. Let us read this question. Consider the following statements. First one, genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. First, let us understand what are the genetic changes. Genetic changes, it means the changes you are doing in the DNA, in the genes. So how you can do it? By using recombinant DNA technology, or other name for this is the genetic engineering. We are having number of genetic engineering tools like CRISPR-Cas9 system, and we are having other molecular seizures and molecular ligation agents that can also be used. So yes, this is true. You can make changes in the eggs or the sperms by using genetic engineering. This is a right statement. A person's genome can be edited. Focus on the word can be. Yes, obviously we can do everything in the given in the statement 1. Can be means whether you are able to do this or not. Yes, we can do it. If we can change any DNA, that is also possible in the case of eggs and the sperm. Only thing is that we need to remove the nucleus from the egg cells or the sperm cells. After that, DNA is the same everywhere. What is DNA? DNA is a polynucleotide. So if you can edit the polynucleotide of a cell, you can also edit the polynucleotide of the egg cells and the sperm cells. So this is only to confuse you. If you know the basics, you can easily understand the main thing behind statement 1. And the coming on the second statement, a person's genome can be edited before birth at the early embryonic stage. Yes, we can do. If you are removing the zygote from the uterus, then you can do it. What is the zygote? This is a fused nucleus. Fused nucleus means after the fusion of the gametes, male gamete and the female gamete. If we are harvesting this zygote, in this zygote we are again having the DNA. DNA again polynucleotide. If I can edit a polynucleotide, why cannot edit another polynucleotide which is present after a certain stage? I can do this also. Again, the term here used is the can be. So this statement is also right. Coming on the third statement, human induced pluripotent stem cells. In 2018, one scientist of Indian origin, Shinya Yamanaka, he received Nobel Prize for this invention. Here the term induced means you are inducing. Pluripotency means tendency to form different types of cells. One cell can form any type of body cell. This is the meaning of pluripotency. It means by using a single cell I can make any body cell. Please focus on the word any body cell. I can make heart cells, I can make kidney, lungs, clear, with a single cell. This potency is known as pluripotency. Now, this pluripotency is present only at the time of birth. In the embryo, we studied the formation of the zygote, how the zygote is produced by the fusion of two gametes, male gamete and the female gamete. Now, this is a single cell. This is a single cell. Now, this cell will multiply. This will make a complete organism. So, by this single cell, we are making all the possible body cells. So, this capacity, this pluripotency is available in the embryo. But this potency will not be there when you grow old, when you become an adult. So, scientists tried to induce this potency, this pluripotency in the adult cells. How they did? They changed the genetic sequence of these adult cells matching with the embryonic cell. After you change the sequence, what is the main thing which controls everything inside a cell? This is the nucleus. Inside the nucleus, basically DNA is controlling everything, and we are making this DNA, the adult cell DNA, matching with the embryonic cell DNA. It means this adult cell will start behaving in the same way as the embryo. So, we can do this. This process is known as induced pluripotency. Now let us look on the third statement. Human induced pluripotent stem cells can be injected into the embryo of a pig. Why cannot we inject this pluripotent stem cell which we prepared by inducing pluripotency in it? Yes, we can do so. It will be beneficial for us. If we are doing this, we can make like the kidney cells, kidney itself, heart itself from the pig. Clear? That can be used for the organ transplantation. This is the biggest hurdle because there is a problem of rejection. When you go for organ transplantation, there is a problem of the rejection. But if I am doing this with the cell of the patient, like I have taken a cell from the patient and I have induced pluripotency in this cell, now this cell can be used to make particular organ which I require to be transplanted in this patient. So if I am doing this, there will be no rejection because the kidney or the other body cells or the tissues which I want to transplant, that will be matching entirely with the patient. So there will be no rejection. So by using third statement, I can do so. So this will be right and this will be desirable. Again the word here can be. So this is also a right statement. Because of this, now we know first, second, third all are right. The right answer will be D. Next question. What is the importance of using pneumococcal conjugate vaccines in India? What are vaccines? Going on the basics. Vaccines are basically the live or dead or some parts of the bacteria or viruses which are injected inside a healthy person. These parts of the bacteria, viruses or itself bacteria or virus, they induce immune response inside the healthy person. So substances which induce immune response inside a healthy person, these are known as the vaccines. Now what is pneumococcal conjugate vaccine? This is a vaccine against pneumococcus, Streptococcus pneumoniae. This is the name of the bacteria, Streptococcus pneumoniae. And PCV, in short, pneumococcal conjugate vaccine, PCV. This is a vaccine against this bacteria, Streptococcus pneumoniae. What does this bacteria do inside your body? This bacteria is responsible for development of pneumonia. Pneumonia is a disorder of your lungs. Pneumonia, meningitis. Meningitis means inflammation of a layer of the brain. Meningitis and other related symptoms. This bacteria results in pneumonia and meningitis. If I am using this vaccine will be preventing me from any future infection by this bacteria. Clear? So I will be prevented indirectly from the pneumonia and the meningitis. This is clear from this much information. Now if I will be prevented by the natural mechanism of my body, how natural mechanism will work here? When I will be given this PCV vaccine, my body will start formulating antibodies, and antibodies, in the antibodies we are having one memory cells, B memory cells and the T memory cells. These memory cells will keep a memory of this infection or of this encounter with the antigen. In the future, when I will be having encounter with this Streptococcus pneumonia bacteria, my immunity will start making the same antibodies which was made during the vaccination. So I will not be requiring any antibodies, I will not be requiring any antibiotics against this bacterial infection, and I will be requiring naturally because of the natural immunity of my body. So this will be the main mechanism by which vaccines work. Now let us look on the different statements. These vaccines are effective against pneumonia as well as meningitis and sepsis. Sepsis means the blood infection. Sepsis means the blood infection. So here the first statement will be right. Now looking on the second statement, dependence on antibiotics that are not effective against drug resistant bacteria can be reduced. Yes, we can reduce it. Reason being, if I am recovering by the natural immunity, why I will be requiring antibiotics? I will not be requiring antibiotics for this. So dependence on antibiotics that are not effective against a drug resistant bacteria. Drug resistant bacteria means the antibiotics will not be active against such bacteria because they have developed resistance against a particular antibiotic. But here we don't require antibiotics, so ultimately dependence will be low. Why we don't require antibiotics? Because we already have the antibodies in our blood, so we don't be requiring any antibiotic. So this is also a right statement. Third statement, these vaccines have no side effects and cause no allergic reaction. This will be wrong. In most of the vaccination programs, there is allergic reactions on the site of administration, and patients will be feeling chills, fevers, pain at the site of administration. The same was true for the COVID vaccine. When the patients were, or the healthy people were given the COVID vaccination, they received vaccination, they felt fever for a day or 2 days. The same thing happens with other vaccines. So this is a wrong statement. The right answer here will be 1 and 2. I think now this is clear, everyone. Now this is our next question. Which of the following statements are correct regarding the general difference between plant and animal cell? See, plant and animal cell, if you go on the basics, in a cell, in a living cell, we are having one nucleus. Basically cells are of two types. I am going on the basics. After the basics I will be taking this question. Cells are of two types, prokaryotic cells and the eukaryotic cells. Pro means primitive, karyos means the nucleus. The cells which are having primitive nucleus, those cells are known as the prokaryotic cells. For example, the bacterial cell. In these cells, we are having a primitive nucleus. In the primitive nucleus, we don't have membrane bound nucleic acid. The nucleic acid is of two types, DNA, RNA, deoxyribonucleic acid, RNA, ribonucleic acid. This DNA, RNA is present directly in the cytoplasm. It is not membrane bound in the prokaryotic cells. Clear? Along with that, we don't have membrane bound organelles inside the prokaryotic cells. Everything is present only into one compartment, and that is the cell, complete cell. When it comes to the eukaryotic cells, we are having U means true, karyos means the nucleus, true nucleus, it means the membrane bound nucleus, and we are having membrane bound organelles present inside the cytoplasm. Now in the eukaryotic cells, on the majority of the level, we divide it into two types of cells. One is the plant cell, second is the animal cell. There is a difference between plant and the animal cell. Plant cells are having one additional layer outside the plasma membrane. That layer is known as the cell wall. Cell wall is made up of the cellulose, hemicellulose, chitin, etc. These are nothing but the organic compounds. These make the outer cell wall of the plant cells. But this cell wall is absent in the animal cell. First difference between plant cell, animal cell. Plant cells are having cell wall, but animal cells will not have cell wall. Both will be having plasma membrane, plant cell, animal cell. There are few more differences between both. In the plant cells, we are having a one large central vacuole. This vacuole is one organelle which is present inside the cytoplasm. In the plant cells, the nucleus is present on the periphery because we are having a one large central vacuole. This central vacuole, it is an organelle present inside the cytoplasm. Vacuole is an organelle present in the cytoplasm. It is responsible for the osmotic balance of the cell. It will provide turgidity to the cell. It will keep the cell in a proper shape. This is the function of this. Now in the plant cell we are having one large central vacuole, but in the animal cells we are having number of small vacuoles. They will be doing the same function in both plant and the animal cell. These are the basic differences between both. Now let us look on the given statements. First statement says plant cells have cellulose cell walls while animal cells do not. Yes, this is a right statement. Now the second, plant cells do not have plasma membranes. This is wrong. How the cell can exist without the plasma membrane? This is the outer layer of a cell. This membrane gives shape to the cell. It keeps everything enclosed. So this is a wrong statement. If you want to go on the answer, even if you have studied only two statements, you can make this question. How? Because second is wrong and it is saying the correct. Second, here also second, here also second. Right answer will be C only. But we are discussing here, let us discuss the third statement. Mature plant cell has one large vacuole. This half part is right. Yes. While an animal cell has many small vacuoles, this is also right. So ultimately the answer will be C. But you can see that even if you are not reading the complete question, still you are getting the right answer. So be attentive in the exam. This is our next question with reference to the solar water pumps. Consider the following statements. Solar water pumps, it means the pumps which are running by using solar energy. This is the meaning of solar water pumps. Now let us look on the statements. Solar power can be used for running surface pumps and not for submersible pumps. Power is power, it has nothing to do. If you supply power to any equipment, it will run. This is like artificial differentiation only to confuse you. So solar power can be used for running surface pumps as well as for the submersible pumps. Now the first statement is wrong. This is like commonsensical. And the second statement, solar power can be used for running centrifugal pumps and not the ones with piston. This is also wrong. Reason being, power is power. You can run any equipment. How it will differentiate? What are centrifugal pumps? Centrifugal pumps are basically, here you will be having revolving part. If there will be centrifugal force generation inside a pipe, then centrifugal force will create a vacuum in the middle of this pipe. And with this vacuum will be allowing the water to be pumped from the bottom towards the top. So this mechanism is there in the centrifugal pump set. And second, piston, you know, the piston, with the help of the piston, you are pumping the water from the bottom to the top. So these are two types. You can do anything with the help of solar power. So this statement is also wrong. And the right answer here will be D. In India, why are some nuclear reactors kept under IAEA safeguards? What is IAEA? It is International Atomic Agency. International Atomic Energy Agency. IAEA means International Atomic Energy Agency. Atomic means it will be dealing with the nuclear atoms. Clear? We are having one more agency. Don't get confused with the International Energy Agency. International Energy Agency deals with all types of energies except atomic energy. We are having one different organization, International Atomic Energy Agency, which deals with the nuclear energy, that regulates this field. How? Basically, let me take you to the historical part of this. When India carried out two nuclear tests, one in 1974, one in 1998, and in 1974 the test was named Smiling Buddha, in 98 the test was named Shakti. After these two tests, we became a nuclear state. We declared ourselves as a nuclear state, and obviously this invited economic sanctions from the West and the European countries. As a matter of those economic sanctions, we were denied nuclear fuel from other countries. Reason being, now we can use those fuels for military purpose, so they did not want to provide nuclear fuel to our country. But in 2005, we had one agreement with the US. That agreement is known as India-US Civil Nuclear Agreement. Civil means we are getting the fuel for civilian purpose. We are getting nuclear fuel for the civilian purpose. Civilian purpose here means for the electricity generation, not for making bomb. Now, as per this agreement, we will be given nuclear fuel only when we will be committing ourselves to the IAEA safeguards. What are these safeguards? According to these safeguards, the IAEA inspectors will be visiting the nuclear reactor sites, and they will be checking that imported nuclear fuel is used for the power generation only, and it is not diverted for bomb making. This is what the present situation. Now, because of this present situation, we are having two different types of civil nuclear power plants. One which are running with the imported nuclear fuel. Those power plants are under the IAEA safeguards. But other category of the nuclear power plants which are using our domestic nuclear fuel, they are not under IAEA safeguards, because we are not using imported fuel. So we don't need to, we are not required to give any justification for that. Now, with this background, let us look on the given statements. Some use uranium, others use thorium. This is illogical. Thorium is not used as a fuel till now. Some use imported uranium and others use domestic supplies. This will be the right answer. For the sake of discussion, I am reading the C statements. Some are operated by foreign enterprises. Why? This cannot be sound logic. Some are state owned and others are privately owned. All are state owned. So the right answer will be B only. Next question, steel slag can be the material for which of the following? First, we must know about the basics of the steel slag. Slag, during the production of the pure steel, we are producing the pure steel from the in a blast furnace. In the blast furnace, basically we want to separate out impurities from the pure steel, and when you separate out impurities from the steel, there will be semi-solid material which will be there at the bottom of the furnace. This semi-solid material, or you can say the byproduct of steel making, is the steel slag. It contains number of impurities, so like the earthen materials and some metals mixed with them. Now it is asking for which purpose it can be used. Now let us look on the statements. Construction of base road. Yes, it is like a mud, or it is like the byproduct. Why we cannot use it for the making the road? We can use it. Improvement of agricultural soil. It contains also some metals and the nutrients, calcium and the phosphate. So it can be used for improvement of the agricultural soil. It can reduce the acidity of the agricultural soil. Calcium oxides or hydroxides are basic in nature, so it can be used for the acidic soils. ultimately you can use it for improvement of the agricultural soils. This is a right statement. Production of cement, yes, we can use it for the production of the cement. Reason being, here we are having number of metal oxides as well as the mud that can replace the Portland cement. So third statement is also right, and the right answer will be D, 1, 2 and 3. This is our next question. Consider the following statements. Coal ash contains arsenic, lead and mercury. See, you will be surprised to know that your coal burning releases some volatile organic compounds, and those volatile organic compounds is having mercury inside it. Though mercury is a metal, and one question may come that this is the only metal which is in the liquid stage. Which is in the liquid state. So yes, coal burning also releases mercury. It will be slightly going against the common sense, but it is also a fact that mercury is one of the ingredient which is released by the coal combustion, and coal is also having certain heavy metals like arsenic, lead and the cadmium. So please remember this fact: coal burning releases mercury, arsenic, lead and the cadmium in the atmosphere. The first statement is a right statement. Second, coal-fired power plants release sulfur dioxide and the nitrogen into the environment. This is also right. Coal burning releases nitrogen oxide and sulfur oxide into the atmosphere. High ash content is observed in Indian coal. Why? This is a true statement. In India, most of our coal reserves are from the bituminous and lignite category. We are having very less coal reserves which belongs to the anthracite coal variety. We are having 4 different coal varieties. Starting with the anthracite going towards the peat, the amount of carbon will reduce. So the amount of carbon will reduce, it means more impurities will be there. If more impurities will be there, then it means high ash content. What is ash content? It means unburnt hydrocarbons. Ash means unburnt hydrocarbons. If we don't have the carbon in the coal, then it will remain unburnt. Unburnt means the amount will be more. And anthracite, bituminous, lignite, peat. These are the 4 different coal varieties, and the amount we are mostly having bituminous, lignite coal. So amount of carbon will be lower in bituminous and lignite as compared to the anthracite. So ultimately it will generate high ash content. So all the 3 statements are right. Answer will be C. Next question. In India, the use of carbofuran, methyl parathione, phorate and triazophos is viewed with apprehension. These chemicals are used as, basically these are chemicals which are used as the pesticides, and the Kerala government has banned all these chemicals. Kerala government has banned all these chemicals. So the right answer is pesticides in agriculture. They have replaced these chemicals with the less harmful chemicals. So please remember carbofuran, methyl parathione, phorate and triazophos. These are the chemicals which are used as the pesticides. The next question, in the context of Which one of the following are the terms pyrolysis and plasma gasification mentioned? First, let us try to understand the pyrolysis. Pyro means the temperature, high temperature. Lysis means the breakdown. It means we are digesting the organic compounds or materials at high temperature in an inert atmosphere. Please remember, when you do pyrolysis, when you break down organic compounds at very high temperature, you do all this in the inert atmosphere. This will result into the solid substances at the bottom along with the release of certain volatile organic compounds. Now, next term, plasma gasification. Plasma gasification means the material which you are getting after pyrolysis, it is converted into the different gaseous molecules. How you are converting this into the different gaseous molecules? By heating it with certain gases. For example, after this gasification process, we are getting a syngas. Syngas is a synthesis gas. What is the chemical composition of this synthesis gas? Hydrogen plus carbon monoxide. This hydrogen and carbon monoxide can be used as a fuel, or hydrogen plus carbon monoxide can be converted into other types of fuel like the methane and the higher hydrocarbons you can make with the help of this syngas, hydrogen and carbon monoxide, which acts as a fuel. Basically, you are doing this with the organic waste collected from the municipal waste, agricultural waste and other biodegradable waste. So, it is a method of waste to energy technologies. Right answer will be waste to energy technologies. This is our next question. Consider the following statements. Some species of turtles are herbivorous. What do we understand by herbivorous? Herbivorous means, in case of marine food chain, herbivorous means which are eating phytoplanktons. These are small plants. So yes, we are having number of turtle species which are dependent on the phytoplanktons in the marine food chain. The first statement is a right statement. Second statement, some species of the fish are herbivorous. Yes, we are having surgeon fish and parrotfish which are dependent on the phytoplanktons. This is also a true statement. Third, some species of marine mammals are herbivorous. Most prominent examples are the manatee and the sea dugong. Manatee and the sea dugongs are the mammals which are herbivorous. These mammals are present in the Gulf of Mannar. This question was again asked in the UPSC. This also gives you the hint why you should focus more on the PYQs. Reason being, not only the same question will be repeated, but the statement of a question can be made in the form of a different question. So always go through the PYQs. It will help you in the UPSC exam. Even if you are not getting good score here in the different test papers, reading PYQs will help you to go through the 2024 exam. Please focus on the PYQs. So this statement, third one, some species of marine mammals are herbivorous. This is also a right statement. Fourth one, some species of snakes are viviparous. What are viviparous? Snakes normally lay eggs, but there are few species of the snakes which give birth to the live young ones. Clear? This is the meaning of the viviparous. The snakes which give birth to the young ones, not the eggs, not lay eggs. And we are having green anaconda. It give birth to the live ones, not the eggs. We are having more species of the snakes like boa, that is also viviparous. So all the given four statements are right. The right answer will be 1, 2, 3, 4. The word Denisova is a place in Scandinavia. It is a place in Siberia. So, in the caves of Denisova, remains fossils were discovered. These fossils belonged to Denisova hominis. Denisova hominis is one extinct human species. Denisova hominis is one extinct human species, and with this information, the option B will be your answer. With reference to recent developments in science, which one of the following statements is not correct? Please look on the word not correct. So, we need to eliminate the statements. Functional chromosomes can be created by joining segments of the DNA taken from cells of different species. This itself is a wrong statement, and this is your answer. If you know the information, you need not to go through all the given statements. How? You can edit the DNA, but till now it is not possible that you have created a functional chromosome by taking one part of a chromosome from one species and connecting it other part of a chromosome with other species. Chromosome is a large molecule. Chromosome is a large molecule. We have not taken chromosomes, we have taken only the DNA. DNA from one species can be merged with the DNA of other species, but this is not possible. Like you have connected two chromosomes and you have created a functional chromosome. This has never happened till now. So, this is a wrong statement, and the question is asking you about the not correct. So, this is your answer, but let me go through the other statements. Pieces of artificial functional DNA can be created in lab. Yes, you can create it. You can create it because we have human genome project. We have understood all the sequences of the genes. Now you can make any DNA sequence in the laboratory by placing one nitrogenous base beside another nitrogenous base. You can create a particular sequence, so you can make one artificial functional DNA. A piece of DNA taken out from an animal cell, it can be made to replicate outside a living cell. Yes, we are already doing with the help of polymerase chain reaction. PCR is an amplification technique by which we amplify DNA. We are already doing this. Cells taken out from the plants and animals can be made to undergo cell division in laboratory. Yes, we are already doing it. Reason being, we don't use only a single cell in the lab. We use a number of cells. Those cells are prepared in the laboratory by artificial multiplication, that is known as the tissue culture. We are providing nutrients to different cells. In the proper nutrient medium, the cells multiply and you harvest those multiplied cells. So, this is all about today's discussion. I am pausing here and we will be taking few more questions in tomorrow's class. All the best and go through all the questions. I will meet you tomorrow.